Cytogenetics, Part 1, Module 1, Lecture 4 in the Genetic Series. Human chromosomes were first identified in 1959. Humans have 46 chromosomes in 23 pairs. 22 are the same, known as autosomes, and the last pair are the sex chromosomes. What they actually are is very tightly wound DNA around histone proteins to compact that information. Here is just a diagram. Here are cells. Inside the nucleus are the chromosomes, and the chromosomes are like a scaffold that have the master copy of all our genetic code, which is used as a template for a protein. So there are inherited chromosomal abnormalities and acquired. We are primarily going to be talking about inherited chromosomal abnormalities in this module. Acquired have more to do with cancer. So why would somebody do chromosome analysis? Well, one is prenatal diagnosis. AMA stands for advanced maternal age. An older woman has a higher risk of having a baby with an abnormal number of chromosomes. Or there are a variety of screening tests, a non-invasive screening test. Multiple pregnancy losses, and a family history of mental retardation, birth defects, or a specific chromosomal abnormality. In pediatrics, we do cytogenetics for children with developmental delay or multiple birth defects, and finally, cancer for acquired chromosomal abnormalities. Tissues, basically anything with a nucleus, blood cells, but they're the white cells, the T lymphocytes. Red cells don't have a nucleus, skin, muscle, placenta, products of conception. Basically, when you have a pregnancy loss, anything that comes out is considered product of conception. And then with cancer, solid tumors, bone marrow. With when someone is pregnant, you can do an amniocentesis, and skin cells from the baby slough off. And then chorionic villi is another prenatal diagnostic test in which a piece of the placenta is taken and the chromosomes are analyzed. 99% of the time, the CVS or the placenta has the same chromosomes as the baby, but 1% they do not.
So
this is just a spread of human chromosomes. Here's actually a cell that didn't break up. And here are the spread, they just kind of randomly spread. Here's one of our chromosome 21s. Here's another one. So here's our 22. And basically the chromosomes, a karyotype is put them in size order. The largest human chromosome is 1. The smallest is actually 21, but at the time they thought in unbanded chromosomes the 22 is bigger than 21, but then they called trisomy 21 Down syndrome, so they never switched it. Girls have 2 X's and boys have an X and a Y. The size of the chromosome, though, doesn't tell us how much genetic information. For instance, the chromosome with the most genetic information is 1, followed by 2, followed by 3, followed by 19. The chromosomes with the least amount of genetic information are 13, 18, and 21. And these are the chromosomes that are most likely to result not in a conception with an extra chromosome, but an actual identified pregnancy or a live born. So it's not that trisomy 21 with non-disjunction occurs more commonly. It just more commonly results in a baby that's born. So we're going to go over a little bit of the anatomy of the chromosome. The ends of the chromosome are known as the telomere. The short arm is known as the P arm, and P in France starts for petite, and Q is the next letter in the alphabet. And the centromere is the middle, and the centromere, we call it the middle, but in most cases it's not exactly in the middle. So, nomenclature, if there's an additional chromosome, for example, Down syndrome, the notation is 47 XX or XY plus 21. If there's a deletion, it's indicated by the arm and the region. So deletion 5, so this would tell us the chromosome 5. P is the short arm, 14.2. When we just see one number, everything above here would be assumed to be deleted. If there's an interstitial deletion, that's in the middle of the chromosome, it would be deletion 5, P, let's say 1.2. to 1, 2.3. So this region here would be deleted. So if you see two numbers in the middle of the parentheses, that means it's interstitial. If you just see one, that's assuming that it's a terminal deletion. Everything above there is missing. Now, gene location has been, this nomenclature has been used for gene location for linkage analysis and whatnot. So when you remember we studied about cystic fibrosis, and the CF gene or the CFTR gene is located on the long arm of chromosome 7, so 7q31.3. And that codes for the gene that has to do with the chloride channel in the cell membrane. Aneuploidy is the most common cause of pregnancy loss. Having an abnormal number of chromosomes, about 50% of first trimester pregnancy losses, and when it says abortices, it really means pregnancy, spontaneous pregnancy losses. The vast majority of them are numerical, and a small percentage are unbalanced chromosome rearrangements, which we're going to talk about in a second. A fetus of mothers over, when we do prenatal testing of women over 35, 1 in 50 have a chromosomal abnormality, 85% are numerical, 10% are balanced, and 5% are unbalanced. But by the time a baby is born, it goes from 50% in pregnancy losses to 1 in 160. And that's because of a lot of these pregnancies are lost. 60% are numeric, 30% are balanced rearrangements, and then another 10% are unbalanced. And so even of this 160, we're going to talk about balanced rearrangements, there's actually no phenotype. But it is technically a chromosomal abnormality. Again, the chromosome aneuploidies that we see in liveborns is the smaller chromosomes 13, 14, 15, 21, and then the sex chromosomes, a loss of the X chromosome in 45X, and extra X in Klinefelters, and then 4p minus and 5p minus are also, for whatever reason, more commonly seen than some other deletions. So we're going to talk about non-disjunction. Normally, during meiosis, we divide our cells in half, half from mom, half from dad, and divide, and we get a healthy baby. Sometimes mistakes happen, and here is an individual that this is chromosome 1, and let's call this chromosome 2. We divide the chromosomes equally, but in this case, both chromosome 1s go to one cell, and the chromosome 2s divide equally. And in this cell, if this is used for fertilization and dad gives, mom gives 2 copies and dad gives 1 copy, the offspring gets 3 copies of chromosome 1 and 2 of 2, or let's say all the others. And in this cell, there's only a single copy of chromosome 1 given by the father, and so there's monosomic for chromosome 1. Monosomy is almost always lethal. The only exception to that is Turner syndrome. Trisomy, as I was saying earlier, is usually lethal, and the exceptions to that is 13, 14, 13, 18, 21, and the sex chromosomes. Over 50% of all first trimester pregnancy losses have an abnormal chromosome constitution. Chromosome analysis should be considered in a woman who's had more than 2 to 3 pregnancy losses, or an older fetus that is lost with multiple anomalies. It's very important to separate the fetal from maternal tissue. And so, for instance, if you were doing chromosome analysis and 100% were normal female, you're probably not doing it right. This is just a diagram talking about the risk of chromosome aneuploidy as you get older. The risk of having a baby with Down syndrome at the age of 25 is about, or it's any chromosomal abnormality at birth, is 1 in 476. At 30, it's about 1 in 385. At 34, it's 1 in 244. At 35, it's 1 in 79, 1 in 179. But it really starts rising in the late 30s and early 40s. But even at 49, 6 out of 7 babies actually don't have a chromosomal abnormality or Down syndrome. Now, why do we offer prenatal testing to women over the age of 35? Well, the original studies on the risk of pregnancy loss due to amnio done in the 70s stated that the risk of pregnancy loss due to amnio was about 1 in 200. And so from this data, why would you offer, 1 in 200. So why would you offer a prenatal test if the risk of pregnancy loss was greater than the risk of finding out anything wrong. And when does that number cross? Well, between 34 and 35. So it's not that a woman 35 has fallen off the face of the earth. And boy, you know, when you're 34, you're not going to have a baby with Down syndrome, but man, you turn 35 and the risk is so high. That's not true at all. And these numbers are very arbitrary. Right now, under ultrasound guidance done by a maternal fetal medicine specialist, the risk of pregnancy loss due to amnio is actually about 1 in 500. So if you use that same statistical information that we used back in the 70s, then you would say everyone should get an amnio, and yet nobody wants to have an amnio. So, and again, advanced maternal age is technically written as over 35, but that's a very arbitrary number, and from the perspective of cytogenetics, it's less and less people are actually having prenatal testing. So, Down syndrome, when a baby has the clinical characteristics of Down syndrome, 95% of the time, there's an extra copy of chromosome 21. So instead of there being 46 chromosomes, there's an extra chromosome. So it'll say 47, XX or XY plus 21. About 2 to 4% of mosaic. And what mosaic means is the conception started out with the extra 21, but that extra chromosome was lost at some point during the development. And then finally, about 2 to 4% are a translocation. An individual with Down syndrome that has a Robertsonian translocation actually only has 46 chromosomes. They don't have 47, but the Robertsonian translocation indicates 2. But if they actually had Down syndrome, then it would have this extra 21 at the end of the nomenclature. So here's an individual who is a carrier for, he has Down syndrome because of a Robertsonian translocation. And so here we have 2 copies of 21. And here's the third copy on top of the 14. So 1, 2, 3 copies of 21. And again, they don't have 47 chromosomes. They have 46 because this counts for 2. But at the end of this nomenclature, it says plus 21. Whereas a carrier of a Robertsonian translocation has 1 chromosome 21 here and the other here. They actually only have 45 chromosomes. But there's a derivative chromosome or a translocation between 1421. And so if somebody had 45XX or XY derivative or translocation 1421, they actually have a normal phenotype. This individual has the right amount of genetic material. Losing the short arms of acrocentric chromosomes, which is 13, 14, 15, 21, and 22, doesn't really hurt you because it's repetitive information. So the long arm of these chromosomes is where all the genetic information is. And so this individual is balanced even though they only have 45 chromosomes. The real problem with a balanced carrier is what do you do when you're having to undergo synapsis during meiosis, prophase of meiosis 1. So how do we line up the 14s and the 21? So you have to make this kind of triangular tetrad here for synapsis. But after synapsis, how do the chromosomes separate? So if the normal 14 and the normal 21 go to one side, leaving the 1421 in the other cell, then when this is fertilized normal, this is fertilized, this is 2 copies of 21, 2 copies of 14, they're balanced. But what if the 1421 migrates with the 21, leaving the 14 alone? In this situation, when this is fertilized, we have 3 copies of 21 and 2 of 14, as is in the karyotype that I showed you in the previous slide. In this case, we have 2 copies of 14 and 1 of 21. What if the 1421 migrated with the 14, leaving the 21 alone? When this is fertilized, we get 3 copies of 14 and 2 of 21, or 1 copy of 14 and 2 of 21. This is actually lethal. This is incompatible with life or lethal. This is incompatible with life or lethal. And the only of these other possibilities that you can end up with a live-born baby is Down syndrome, which is because of an unbalanced translocation because of the incorrect separation. So in the real world, if a parent is a carrier of a Robertsonian translocation, the chance that they will have an offspring with Down syndrome is not like 80%, it's 15%. So here's just a diagram of the separation, and here our chromosomes are separating. But here we have a Robertsonian translocation, so in alternate segregation, we get our 1421 in one cell and our normal 14 and 21 in the other cell. But in adjacent, if the 1421 migrates with the 21, leaving the 14 alone, when this is fertilized, this will result in Down syndrome, and this will result in a monosomy. So if a parent carries a 1421 translocation, there's a 5 to 15% chance of having a baby with Down syndrome. If the baby is born with Down syndrome and found to have a 1421 translocation, however, there's only a 50% chance that the parent carries it. About 50% of the time, it was something new in the egg or sperm. So there's a difference between, and only, like I said, 2 to 3% of babies with Down syndrome have a translocation. So a baby has chromosome analysis indicating 46XY translocation 1421 plus 21. What is the diagnosis? Normal male, normal female, male with Down syndrome, or female with Down syndrome. And the correct answer is male with Down syndrome. That concludes this module.